Welcome, everyone, and thank you for joining us for today's webinar, General Informatics Strategy for Specific Scientific Data, From Collection to Insight, presented by Dr. Johan Potier, Technical Associate Director, Novartis Institutes for Biomedical Research. My name is Xavier Gutierrez, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Potier. I will now turn the presentation over to him.
Thank you, Xavier. Good afternoon, everyone. So today we're going to speak about how we handle our data at Novartis and while working with particular scientific needs. So just a brief overview of the agenda. I'm first going to speak about my organization, how we approach data management and why, and I will finish by speaking about architecture and our data mining goals. So for those of you who don't know, Novartis is a pharmaceutical company, and the Novartis Institute for Biomedical Research is the research branch of Novartis. We're about 6,000 people worldwide across 6 campuses, the main one being in Basel, Switzerland, Cambridge, US, and Shanghai, China. So I'm currently located in Cambridge, US. We collaborate across scientific and organizational boundaries with a focus on new technologies to produce therapeutic breakthrough for patients. So we focus lately a lot on collaboration with academia and other smaller companies to develop new ways to interrupt cellular machinery causing disease. And the framework that our president, Jay Bradner, put in place is as open source as possible with open collaboration being the key word. So you can see on this NIH graphic the drug discovery process for small molecule and biologics from early research on the top left corner down to clinical trial and marketing down at the bottom of that graphics. I realize here that the font is very small, so don't worry too much about it. What's important is we focus on research and target identification at the institute here. And you can see already on the screen that there is some informatics component to that research with data mining, data repository, and biorepository. So this is where the organization that I'm part of is helping the scientists. We support the scientific community. We support them by providing an infrastructure, recently more and more in the cloud. We provide them with data pipeline capabilities, data integration, analytics, and automation. All this really to help the drug discovery process and facilitate the development and the deployment of application. So it's obviously tempting to speak about data science and machine learning right away. But before that, the focus remains on supporting the users and their workflow. It also forces us to evaluate the way we record data, the one generated by the users, but also the metadata from the instrument and the various devices that are being used in the organization, or the one we receive from external partners like CRO. In this pyramid, in this chart, you can see a good representation on the needs and dependency we need to address before working with AI. It can be addressed for each project, for very specific needs, but also working at the organization level. So it really requires initial work, and this is what I'm going to address today. You can see at the base of this pyramid that collection of the data is the initial step, from instrumentation, user data, sensors, partners such as CROs, and it can be as sophisticated as your solution is, and the data needs to be captured and stored. The way you store it is relevant. How do you provide that data to your pipeline? Where is it stored? Do you need to transform it? For example, do you need to normalize your raw data for analysis? And what kind of infrastructure you need or want to have for your particular type of data. And you can see as you're going up in the pyramid, you're focusing first on the collection, how to store it, how to explore that data, and finally you start addressing the analytical needs with aggregation, optimization, and finally AI. But before going all the way up to the top with deep learning, machine learning, and other AI techniques, you need to address the bottom of that pyramid. This is by no means mandatory, and it's not always required depending on what you do, but this is how we address it here at Novartis, and how we decided to address the data challenge. While I speak about where we want to go, we still face many challenges, and I'm sure other organizations have the same problem, coming from years of legacy data and a less data-centric world. Organizations are not historically organized around data, so that's really the first hurdle that people encounter. You usually create silos around functional organization, around the type of application that you're using, and it's really difficult to work cross-functionally. Decision around the governance of the data is also complicated. People that do the analysis might be the owner of the data, but the requester might be the one doing the analysis. How do you handle it will really cause issues down the pipeline if you decide to do data analysis. Infrastructure can also be very difficult. Tools are not always the same for all the teams. Depending on their focus, they will have different version, different type of tool, different automation, and it makes it really difficult to share data across the entire organization. And of course, people have very different background. They see different aspects of a particular problem, and they usually focus on their own goal rather than thinking globally about data. All these different challenges we are facing now, some of them are being addressed and some will need to be addressed later on. Overall, we spent a huge amount of time and resources to really do simple analysis. It's a lot of manual steps. There is no common ontology, common vocabulary across the organization. So the way we do analysis is not consistent, not scalable, and there is really nowhere centralized to share data in a simple way. The data being handled is of course also quite difficult because it's very diverse in its nature. You see here a few examples that I chose for the audience. The source of the data is different, the type of the data, the size of it. Everything is not structured correctly. You can see images here with the high content screening example, animal data, registration of different type of entities, location of your sample, of your animals, request tool and communication with external partners for procurement, for example, are another case. And of course, you have the pure scientific data associated with each disease area. A neuroscientist will not explain the way they did a particular electrophysiology experiment the same way that a cardiovascular specialist will. And this type of communication should be addressed while working on the way you're storing the data. Here's a simple example. In an in vivo case, for example, how we could collect data. Very diverse type source of information. A tissue sample here will be associated with a particular tumor model. That tumor model can come from an animal that is chipped or has a barcode. This sample relationship between the animal, the tumor model, the tissue that you're using for your experiment need to be tracked and prepared. How do you track that information will depend on how the organization stores the information. And while you're performing your experiment, you might want to have a particular ID associated with, let's say, the antibodies or the small molecule that you're using on your animal or your tissue, and the data that you will have as an output of that study, will it be efficacy data or PK/PD data, will really relate to that original tumor model and tissue management system that you're using. In the end, it might be the scientist directly doing the analysis of that experiment, or it might just store that information into an electronic notebook, and an analyst will do some visualization work, for example, here with Spotfire, but it could be really any type of other visualization tool. Other example can cover the genomic or proteomic profile of the animal, in vitro data, histopathology data, biologics, where is that antibody is coming from, for example, where is the animal coming from, so the procurement data, the type of genotype, and which vendor did we use. In the end, as an analyst, you may want to have access to all that information across the entire pipeline of your experiment to make sure that you're not making any mistake when making assumption and conclusion based on that experiment. All that data has metadata and has to be stored according to its specificities. The data pipeline change depending on the scientific workflow, and downstream operation can be applied to it. Of course, microscopy image, for example, videos, NGS dataset, they obviously have an impact on the storage. This very large dataset needs to be stored in a specific way, and that should be considered in terms of cost, retention, and access. At Novartis, the current approach is actually to implement a data store, commonly called data lake as well. We provide a data catalog that's used to register and find the data easily and fast. Security is present in terms of authentication, so who is accessing the data, and authorization, who is actually allowed to access the data. And all this is in the cloud right now to be scalable based on demand from the users. Exploration is the next step in that pyramid that we saw earlier. You need to be able to clean the data, curate it, make sure that you have a standard way of describing the same properties and the same metadata. Defining that ontology is key and really difficult. It really requires scientific knowledge and really subject matter expert in a particular domain, making sure that when you're describing properties associated with an in vivo sample in cardiovascular, for example, or in oncology, you're talking about the same thing if they can be compared. If they cannot be compared, then you have to make sure that you're describing the namespace or which type of data is coming from oncology, which one is coming from cardiovascular. Something that's coming up quite a lot in the last 10 years is the FAIR principle. Data and supplementary material have to have sufficiently rich metadata and unique and persistent identifier to be findable. That's really key to find your data after storing it. And I do not mean right after the experiment. I also mean in the next few years, when you want to relate to it, search to see if somebody ran a similar experiment with the same type of condition. It has to be accessible, meaning that it has to be understandable by human and machine. It has to be deposited in a trusted repository. That's really important. Finally, the last 2 points are a little less important in terms of priorities, but they're very relevant to what we want to do on top of the pyramid. You have to use a formal and shared and broadly applicable language to represent your knowledge. I'm going to speak a little bit about the entity type catalog on the next slide, And that's really key here to be able to share a common language in terms of what you're comparing. The last point is reusable. Data and collection have a clear usage license and really provide accurate information on the provenance of the data. That means that when you look at it, you have to be able to track down where is that data coming from, where is the relationship between the different samples, and make sure that you can relate the result to that data set accurately. Again, having an API, meaning programmable access to that data, allow the rest of the organization to leverage specific information, and that's really key to adoption by your organization. I mentioned the entity type catalog earlier. This really allows you to describe your data model accurately. We took a very simple approach by describing everything either as a sample or an object related to a protocol and having a data set as a result. For example, this is a very simple in vitro experiment where you have a sample here represented in orange. That sample has to have an ID. It is required for a particular activity. As shown on the right side, it could be a type of assay, a protocol instance, and that sample that could be either a molecule, a protein, a plant extract, or so on, produce a particular data set. It can be a result, a readout of a particular instrumentation related to that activity. So you see here in that simple graph that that data set can be directly linked back to the sample with its own identifier. And you can cover more complex activities and model all your scientific workflow in a very consistent framework that allow you to really compare apples to apples and orange to oranges. In terms of technology, our current solution is really to build platform and web services. Small microservices apply to very specific needs that talk with each other. That way we can really develop them really fast. They are easy to retire and replace, especially upgrade depending on the needs. They are cheap to maintain. They can capture fair data if they've been programmed that way, and they're really painless to migrate from one type of server to another type of enterprise backend service. That command service layer can be used by applications that we develop inside the informatics department, but they can also be used directly by the bioinformaticians that are embedded with the different scientific team or by external collaborators if needed. Again, that entity type catalog enforces the first principle and enforce the way we describe each entity and their relationship. These relationships are key to find samples associated with particular data set and the relationship between different types of samples. Think again about the example of the tumor model and the animal providing that tumor model. Back to really a little more information about the architecture. I realize here the graph might be small, but what's important is you see on the right side the application used by the users or the bioinformatician. In the middle here in orange, you see the service layer hosted in the cloud in our case. You have these three main services managing activities, sample, and result or data set. And they can communicate directly with the data lake or the registration system where we have identifier from the sample and data set for the result. That service layer can then be interrogated directly by bioinformatician or used by the scientists for analysis in Spotfire or such tool directly without having to address the need directly in the application. So you can have very specific application depending on the need. And if I take back the example on oncology and cardiovascular, you could have a front end, an application for cardiovascular, storing the same type of information as oncology with a very different user interface. That could depend on the type of automation they use, the type of experiment they use, but that service layer will interpret it as the same type of information, the same type of data, and store it in the same way. So one can compare these two experiments with one another when it is relevant. All this without disturbing the specific needs of each scientist. Here I added this laptop at the bottom, showing that bioinformaticians can perform their analysis either directly on the dataset, directly from the data lake, or can leverage the service layer themselves to look at relationship or try to interrogate the data in a very similar way as the scientists will do. What's important really here is to support the scientific workflow that has been the primary mission of the informatics department from the beginning, while supporting their instrument integration so we can automate as much as possible. We store heterogeneous data, really images, videos, genomic data, screening data, high throughput data, and so forth. But we allow the user to visualize their result and analyze their data in a very consistent way, independently on the type of data and what kind of instrument they're using. The goal being to answer scientific questions. The data is rich, okay, you have high content data and annotation, but they shouldn't be limited by the current tools. They want to be able to access that data directly where it is stored, independently of how they store it. And the analysis goes together with the assay and the method development. So depending what they want to store, how we store it, we can make this search afterward independently of how it was done upstream in the process. The main goal being to search and mine the data itself across application, across disease area, across labs and team. You want to search internal and external data in the same way. It doesn't matter if the experiment was performed by a CRO or a CMO or internally. That data should be stored in a very similar way internally and can be searched by the scientist. Throughout the organization, that data is really rich and important for the future of the research. So it should not be considered as only silos of data according to the teams, but really as a global data source. And for this, it's really important to agree on an ontology and an architecture, how to access it, document everything. Having an API available is really key for adoption by bioinformaticians and the rest of your IT teams. And I will insist on that. The UI on how the application can be used, how the application looks like, will help with adoption. If the scientists do not want to capture the data and continue capturing it in Excel file or their own private tool, that data is lost to the rest of the organization and remain in a silo. So working on how the users interact with the application and how they store the data is key to success. I'll finish on speaking a little bit about data management. To make it work long term, you really need to understand the type of data you're dealing with. That means that you need to work with the scientists, with people in IT that understand the type of data you're dealing with and how to store it. That scientific understanding is key to store the data correctly and to ask the proper question in the end. The generic architecture and how you want to deploy that architecture is key to make it generic and adopted by the IT and by the bioinformatician community. And the data model will help you with that. Finally, data ingestion and integration across the organization will allow everybody to see the added value of having this generic architecture and data management strategy. Every time you add new data source to your existing architecture, you will need domain expert to help you understanding the type of data you're dealing with, its size, and so forth. All this again to address the base of that pyramid, the collect part, then the store part, and the explore. Understanding the scientific data and understanding the scientific problem will be necessary before going further. The goal remains to answer the scientific question, and this you require multiple skills. You don't need only the IT, you don't need only the scientists, you you need to have collaboration between the two departments, and you need to be able to speak the same language. With this, I thank you for your attention, and I'm waiting for your question. Thank you.
Thank you, Dr. Potier, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. And before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May of 2019. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.